Welcome to the Deep Dive. Today we're plunging into the unbelievably intricate world inside our cells. Imagine for a moment a bustling, microscopic city. Every single structure, every process, building new components, responding to invaders. It's all driven by this constant flow of information. And at the heart of this cellular communication network are what scientists call protein-protein interactions, or PPIs.
Yeah.
Think of it as how the workhorse molecules of our cells, proteins, how they talk to each other, forming countless, maybe fleeting, maybe firm alliances to get things done.
That's right. These conversations, these PPIs, they're absolutely critical, essential for almost all cellular signal transduction events. That's how cells receive and respond to messages from their environment. And also for transcriptional regulation, which is, how genes are turned on and off. They truly play integral roles in just a huge range of biological processes. Without them, our cells, they just wouldn't function.
Okay, so our mission for you today is to really unpack this cutting-edge strategy scientists are using to literally eavesdrop on these interactions, especially right inside living cells. Understanding this molecular dialogue, it's like a direct shortcut to understanding disease and, crucially, developing new drugs.
Indeed, because when these protein conversations get messed up within their complex networks, cells can start behaving strangely. And that can ultimately kick off all sorts of diseases. For a concrete example, consider the interaction between PD-1, that's programmed cell death protein, and its ligand, PD-L1. Now, this isn't actually a good interaction. It promotes cancer progression. It helps tumor cells escape our immune surveillance, basically lets them hide.
Right, hide from the body's defenses.
Exactly. But here's the powerful part. By targeting this pathway with inhibitors like nivolumab and semiplumab, doctors can actually turn the immune system back on to fight the cancer. And that's shown significant benefits for some patients.
But here's the challenge then. Studying these interactions, especially the weak or transient ones, the sort of fleeting whispers rather than loud pronouncements, doing that in their native living state is incredibly difficult. Traditional methods, well, they often require taking cells apart, disrupting the very cellular environment we're trying to observe.
Yeah, and you can lose the very interactions you're looking for that way.
Right.
Traditional methods like co-immunoprecipitation and pull-down assays, they can certainly identify lots of protein candidates and quickly. But like you said, they require disrupting the native cellular environment, and they can easily miss those weak or transient interactions. They're really important ones sometimes.
Then there's the yeast-2 hybrid system. Now, that can characterize PPIs in more physiological conditions, but it really only suggests potential interactions, and it can suffer from, well, negative interference or false positives. Makes it harder to confirm what's really happening. So how are scientists overcoming these hurdles? How are they managing to peek into these microscopic conversations in real time, without disturbing the speakers? That's what this deep dive is all about.
Okay, let's start with a really clever approach. Methods that use light to, see interactions. The true beauty of these techniques is their ability to provide, real-time monitoring of what's happening inside a living cell, almost like a microscopic video camera.
Yeah, a prime example is the fluorescence fragments complementation assay, often just called BFC. Its core principle is quite ingenious, really. You take two split, non-functional fragments of a fluorescent protein, and you attach each one to a different protein you're interested in.
Okay.
Then, if your proteins interact, if they come close enough, these fragments reassemble. They become functional again and emit a fluorescent signal.
So the light turning on tells you they've met.
Exactly. It shows an interaction has occurred. And the true brilliance here, it seems, isn't just seeing if they interact, but it's about witnessing the living choreography, right? Understanding not just who talks to whom, but where in the cell and when these critical conversations happen in real time. That dynamic view must be a game changer.
Absolutely. BIFC has already yielded significant insights. For instance, it's been used to study alpha-synuclein and tau interactions. Those are proteins involved in Parkinson's and Alzheimer's diseases. Studying them in rodent models could really facilitate drug screening efforts.
Wow.
And it also helped analyze the interaction between HIV-1 integrase and a key cellular cofactor protein right in live cells. Opens up new avenues for anti-HIV therapies.
And I gather researchers are constantly improving BIFC, making it even more powerful, like using different fluorescent proteins, maybe smaller ones, more stable ones.
That's right. They're developing things like phytochromes, IFP 2.0, for instance, for greater stability. Or using much smaller fragments, like Mir FP67 and Nano, which is way smaller than standard GFP. That means higher photostability and potentially fewer adverse effects on the proteins themselves, less interference.
Makes sense. You don't want the tag messing up the interaction you're trying to watch.
Precisely. And BIFC can even be combined with other techniques. You can simultaneously image multiple PPIs, or even up to 4 distinct proteins. Think about studying multiple components of a pathway, like G protein couple receptors and their partners, all at once.
Okay, but every breakthrough often comes with its own set of limitations, doesn't it? What are the main drawbacks with BIFC?
Well, a major challenge is that those fluorescent fragments, they can sometimes spontaneously assemble on their own, even without the target proteins interacting.
Leading to false positives.
Exactly. Researchers are developing ways around this, like systems called BAC-BIFC. But it's something to watch out for. Also, BIFC isn't ideal for observing really dynamic changes. The fluorescent complex forms quite slowly. The half-time is around an hour.
An hour? That's a long time in cell biology.
It is, and its association is irreversible once formed. So you can see if an interaction happened, but it's hard to see how it evolves over, say, seconds or minutes. You miss the quick on-off stuff.
Okay, that leads us naturally to another category of light-based techniques then. Resonance energy transfer, or RET. I think there are 2 main types, FRET and BRET.
That's right, FRET and BRET. The general principle here is energy transfer occurs when 2 specifically labeled molecules are in very close proximity, typically within about 10 nanometers. But unlike BIFC, they don't necessarily need exact physical contact for the energy transfer to happen.
Which could be an advantage. Less chance of the labels themselves getting in the way.
Potentially, yes. It can reduce steric hindrance.
Okay, so let's dive into FRET first. Fluorescence resonance energy transfer. My understanding is this relies on non-radiative energy transfer, meaning like no light is actually emitted from the donor to the acceptor.
Correct. It's an energy handoff, not light emission. Here's how it works. You have 2 interacting proteins, and each is tagged. One with a donor chromophore, one with an acceptor chromophore. When they come close, due to the protein interaction, the activated donor transfers its energy directly to the acceptor. And that causes the acceptor to emit its own fluorescent signal.
So donor gets excited, passes energy, acceptor lights up. No interaction, no acceptor light.
Precisely. And the power of FRET lies in its ability to measure interaction strength. You can get quantitative data, like the precise bonding between GROEL and GROEs in bacterial protein folding. It can even quantify dissociation constants, how tightly proteins bind over a range from millimolar down to nanomolar.
That's incredibly sensitive.
It is, and when you combine it with techniques like fluorescence, lifetime imaging microscopy, or flow cytometry, you get quantitative data directly from live cells. And there's even single-molecule FRET, SMF-tree.
Right, pushing the boundaries even further.
Exactly. It lets you detect the behavior of individual molecules in real time, overcomes some spatial resolution limits, and gives us incredibly granular insights into single protein events, like figuring out differences between certain chaperones in E. coli.
So FRET sounds amazing for precision measurements.
Yeah.
But what are its challenges? Continuously shining light on cells, that can't be good for them long term, can it?
You're right, that's a key limitation. FRET is susceptible to irreversible photobleaching. The fluorescent tags just stop working after a while. And also cytotoxic photodamage. You're essentially cooking the cells with light if you're not careful.
Yeah, not ideal for long-term observation.
Definitely not. But scientists have developed clever solutions. There's something called dye cycling, which involves reversible dye binding and photoswitching FRET, or PS FRET red. This requires less light, and the probes can be reactivated.
So you can turn them on and off.
Sort of, yes. It allows for repeated monitoring of dynamic processes and gives a much better signal-to-noise ratio. It's been used effectively for monitoring things like caspase-3 activity. That's a key enzyme in programmed cell death.
Okay, so that addresses some of the phototoxicity. Now, what about BRET? Bioluminescence resonance energy transfer. How does that differ?
BRET uses a different light source. Instead of external excitation light, it uses a luciferase enzyme, like the kind found in fireflies, fused to one protein, and a fluorescent protein fused to the other. The luciferase performs an enzymatic reaction using a substrate and emits its own bioluminescence. It makes its own light.
Okay, so no external laser needed.
Right. And then, if the proteins are interacting, that bioluminescence energy is transferred non-radiatively, just like in FRET, to the nearby fluorescent protein acceptor, which then emits its own fluorescence at a different wavelength. That's the signal you detect.
So the big advantage is avoiding the external light source.
Exactly. If we connect this to the bigger picture, Brett is really attractive because it integrates RET's advantages, sensitivity to distance, real-time monitoring, but it ingeniously avoids the phototoxicity and photobleaching caused by external excitation light.
Because it generates its own light source internally.
Precisely. Less stress on the cells, better for longer observations. Common luciferases include ranitidluciferase, or improved versions like RLU8, and there's a newer one, nanoluciferase, or NULUC. It's much smaller, only 19 kilodaltons, and produces an intense sustained signal. This allows for quantification of even very small numbers of interacting proteins, like insensitive caspase sensors.
Okay, this is fascinating. These light-based methods are great for visualizing interactions we kind of already suspect or know about.
Yeah.
But here's where it gets really interesting for me. What about discovering completely new interactions? Especially those weak or transient ones that are so hard to catch. It feels like trying to find a needle in a haystack, but the needle keeps, vanishing.
That's a perfect analogy, and that's exactly where mass spectrometry-based proximity labeling strategies come into play. These methods are specifically designed to identify unknown PPIs, even the weak or fleeting ones. The core idea is to chemically convert those dynamic, non-covalent interactions into stable, covalently linked protein complexes. Basically, you stick them together permanently.
You freeze the interaction chemically.
Exactly. These stable complexes can then be isolated and efficiently analyzed by mass spectrometry, which is incredibly powerful at identifying proteins. So you essentially get a list of all the proteins that were close enough to your protein of interest to be considered interacting partners at that moment.
Okay, so how do you actually do that labeling? How do enzymes help us tag these nearby proteins?
The principle involves using specialized enzymes. These enzymes are fused to your protein of interest, your bait protein. Then you add a specific substrate. The enzyme converts this substrate into a chemically active reactive substance. This active substance then diffuses, but only a very limited distance from the enzyme.
And so it only tags things really close by.
Right. It reacts with and tags nearby proteins with a unique chemical label, usually biotin. These labeled prey proteins are then fished out and identified by mass spectrometry.
Let's break that down. You mentioned peroxidases first.
Yes, horseradish peroxidase, HRP, was an early tool. It's known for its fast reaction kinetics, which is good, but it has limitations. It needs an oxidizing environment to work properly because it relies on disulfide bonds, so it's mostly useful in secretory pathways or on the cell surface.
So not ideal for looking inside the main body of the cell, the cytosol, which is a reducing environment.
Correct. That's where Apex came in. Apex was engineered from pea or soybean peroxidases. It's a monomeric peroxidase, and crucially, it's active in the reducing cytosolic environment.
Ah, so that opened up the inside of the cell.
It did. For example, Apex was used to characterize the proteomic microenvironment, all the proteins nearby the neuronal and lysosomal network. It helped identify new lysosomal proteins.
But I heard Apex had sensitivity issues initially.
It did. The original Apex wasn't super efficient, so Apex 2 was developed through directed evolution, basically, lab-based evolution. Apex 2 offers much higher catalytic efficiency. It labels things much faster and more effectively.
And it's been used for a lot more applications then.
Oh, yes. Elucidating microprotein-protein interactions, identifying glycan-binding proteins. And because it labels so rapidly, it's applicable for profiling proteins in specific, even difficult structures like the insoluble nuclear lamina. There's even a split Apex version for targeting highly specific subcellular regions.
But using peroxidases? Even Apex? It sounds like there could still be a downside. You mentioned HRP needing certain conditions, but Apex uses hydrogen peroxide, right? H2O2.
That's the main potential drawback, yes. You need to add hydrogen peroxide for the reaction to work. And H2O2, even at low concentrations and for short times, inevitably causes some cell damage and can disturb redox-sensitive pathways, even if you add quenchers to stop the reaction quickly.
So it's powerful, but maybe not perfectly gentle on the cell.
That's a fair assessment. Which leads us to the next class.
Right, the biotin ligases, how do they work?
Okay, so EOA is a natural E. coli enzyme that attaches biotin, which is vitamin B7, to a very specific protein sequence. But researchers found a specific mutation, R118G, that creates a mutant version, now famously known as BioID. This BioID mutant loses its strict substrate specificity. It becomes promiscuous, meaning it generates an activated biotin intermediate biotin oil 5-out AMP, which is reactive but unstable. If it doesn't find its normal target quickly, it diffuses away from the BioID enzyme and reacts with any protein that happens to be nearby, specifically attaching biotin to lysine residues.
Ah, so it labels its neighbors indiscriminately.
Pretty much, within a certain radius, yes. And this was used, for example, to study proteins that track along the ends of microtubules, those cellular highways.
Okay, so BioID could label nearby proteins, but I remember hearing it was quite slow.
It was. The original BioID required very long labeling times, typically 15 to 18 hours.
Hours. That's a huge window for things to move around or non-specific background to build up.
Yeah. Exactly. So BioID2 was developed later. It's smaller and designed for more selective targeting and enhanced labeling efficiency. It was used to investigate the cardiac dyad proteome in living heart cells. But even these ligases can be relatively large. BioID itself is 35 kilodaltons. Fusing that chunk of protein onto your bait might interfere with its normal function or location.
Yeah, that raises an important question. How do scientists mitigate that size issue?
Well, one clever solution is using a combined system like the FKBP-FRB dimerization system, coupled with BioID. Here, a much smaller protein tag, FRB, only about 11 kilodaltons, is fused to your protein of interest. The BioID enzyme is separate. Labeling is induced only when you add a small molecule, like a rapamycin analog, which brings the BioID close to the FRB-tagged protein, gives you temporal control.
More precise timing.
Right. And more recently, other engineered ligases like ARID have been designed. ARID shows high biotinulating activity and good proximity dependency. It's proven useful in animal models and even plants. For example, identifying drug-induced neosubstrates of an E3 ligase called cereblon.
So, putting it together, what are the general advantages of these biotin ligase methods compared to the peroxidases like Apex?
The main ones are, they don't require potentially toxic reagents like hydrogen peroxide. They use biotin, which is highly soluble and generally non-toxic, as the substrate. And they utilize intracellular ATP, which is readily available inside the cell, so generally considered gentler.
But the limitation was the speed initially.
Yes. The long labeling time for conventional BioID was a major drawback, increasing background signal. That's what led to the development of Turboid and Mini-Turbo. Turboid is still 35 kg, but Mini-Turbo is smaller at 28 kg. And crucially, they allow efficient labeling within just 10 minutes.
10 minutes. That's a massive improvement from 18 hours.
Huge difference. Turboid, for instance, was used to identify high-confidence interactors of the Lassa virus polymerase that could potentially aid antiviral drug development.
Any downsides to Turboid?
Well, its high activity can sometimes be a double-edged sword. It can sometimes lead to low-level labeling even without adding extra biotin, just using the cell's endogenous biotin. That can make precise timing control a bit tricky sometimes.
And I'm guessing the split strategy we saw with peroxidases also applies here to get more specificity.
Absolutely. Split BioID and split Turboid have been developed. Similar principle. Two inactive halves are fused to potentially interacting proteins. They only reassemble and become functional upon specific protein contact, or sometimes even triggered by light.
Making them really convenient for controlling the enzyme activity.
Yes, and for studying specific intracellular regions, like where organelles touch each other, with potentially fewer false positives because the enzyme is only active right at the interaction site. They've even been used effectively to map the complex interactions between the SARS-CoV-2 virus proteins and human host cell proteins, giving vital insights into how the virus works.
Fascinating. Okay, let's connect this to another related area. Ubiquitin-like ligases. How do they fit in?
Right. These are methods like NAD dilator and PUPIT. They are derived from the cell's own machinery for ubiquitination, or processes like it. Ubiquitination is how cells normally tag proteins for degradation or other fates. These methods hijack parts of that machinery for proximity labeling. NAD dilator, for instance, allows the transfer of a small ubiquitin-like protein called NADD8 onto nearby prey proteins. It's been useful for identifying substrates for E3 ligases, which are key players in ubiquitination, and also for finding proteins that bind to small molecule drugs, like the cancer drug dasatinib.
And PUPIT. Sounds intriguing.
PUPIT uses a bacterial enzyme, a PUP ligase called PAFA. You fuse PAFA to your protein of interest. Then PAFA attaches a prokaryotic ubiquitin-like protein called PUP onto lysine residues of nearby proteins.
So similar principle, different tag.
Exactly. It's been used to identify partners of critical immune cell receptors like CD28, which is needed for T-cell activation, and also to explore the roles of a mitochondria-associated ubiquitin ligase called MRCH5.
And what's particularly exciting about PUPIT, I read, is that the entire system can potentially be expressed within cells, which holds great potential for use in living animals.
That's a key advantage, yes. The potential for in vivo applications. A challenge initially with PUPIT was that the PUP tag itself is relatively large, which could affect labeling efficiency or perturb the system. But researchers have found that shorter versions of PUP, even just the last 26 amino acids or maybe even just 14, can maintain efficiency while offering more flexibility.
And there's a newer version, PUPIT2.
Yes, PUPIT2 reverses the strategy slightly. The bait protein is fused with the smaller PUP tag instead of the larger PAFA enzyme. Seems to work well too.
So overall, what's the benefit of these ubiquitin-like systems compared to, say, BioID or Apex?
Generally, they tend to have lower nonspecific backgrounds. This is thought to be due to higher specificity in the tagging reaction and potentially a narrower labeling radius, because the active chemical intermediates involved might not diffuse as freely as, say, activated biotin.
So potentially a cleaner signal, fewer bystander proteins getting tagged.
That's the idea, yes. More focused labeling.
Okay, this brings us to our final category of these really cutting-edge methods, photoactivation-dependent proximity labeling. How does light play a role here beyond just, activating a fluorescent tag like infrared or BIFC?
Right, here light is used differently. It's used to directly generate highly reactive free radicals or other chemical intermediates, similar in function to what the enzymes do in the methods we just discussed. These light-generated reactive species then react with nearby proteins, covalently tagging them.
And the advantage?
The big advantages are potentially very high spatiotemporal resolution. You can control exactly where and when the labeling happens just by shining light. Plus, it often uses biocompatible conditions and can offer enhanced selectivity.
Let's break that down. First, photosensitizer or photocatalyst-mediated labeling. What are photosensitizers doing?
Photosensitizers are molecules that, when excited by light, generate reactive oxygen species, or ROS, like singlet oxygen. Singlet oxygen has a very short lifetime, maybe microseconds, and a very limited diffusion radius in cells, just nanometers.
Extremely localized.
Extremely. These ROS then oxidize nearby amino acid residues on proteins, particularly tyrosine and histidine, essentially tagging those proteins as being close to the photosensitizer.
And can we genetically engineer cells to make these photosensitizers right where we want them?
Yes, that's a powerful approach. Genetically encodable photosensitizers like killer red or minisog can be expressed as fusion proteins attached to your protein of interest. So the ROS generation is targeted right to the bait protein's location. For instance, minisog fused with an FBOX protein called SKP2 was used to capture its interacting partner, SKP1, just by illuminating the cells with blue light.
But they're also small molecule photosensitizers, right? So you don't necessarily have to genetically modify the cell.
Small molecule photosensitizers offer flexibility. You don't need to introduce exogenous genes. And you can often target them to specific cellular compartments using a targeting moiety, like attaching DBF HOCHT to target nuclear proteins, or DBF with triphenylphosphonium to target the mitochondrial proteome, lets you probe specific locations.
Okay, and then you mentioned metal catalysts. That sounds very chemical, very cutting edge. Ruthenium and iridium.
Absolutely. These are coordination complexes, often based on ruthenium or iridium, that act as photocatalysts. When illuminated with light, they can generate other types of reactive species. Ruthenium catalysts, for example, can generate phenoxyl radicals that label tyrosine residues on nearby proteins. While some early methods maybe involved harsh reagents, researchers have developed much better strategies now, like using ruthenium bipyridyl complexes conjugated to peptides that target specific proteins like MCL1, or using cell-permeable ruthenium complexes to selectively target intracellular proteins, even in intact red blood cells. Some even have dual applications for labeling and protein knockdown.
So what's the buzz around iridium then? Why is that gaining popularity?
Iridium-mediated labeling is really taking off, largely due to its potential for extremely high precision and efficiency under biological conditions, often using visible light. For instance, specific iridium catalysts have been used for profiling mitochondrial proteomics in living cells with really impressive spatiotemporal precision. And another very powerful strategy is called micromap, or MAP. This uses an iridium catalyst to activate tiny chemical groups called diazarines using just blue light. The key here is that the carbene generation, the reactive species, is localized to within perhaps 0.1 nanometers of the catalyst.
Wow, that's incredibly close range.
It is, which leads to minimal background labeling. It shows great potential for identifying antibody targets or dissecting signaling pathways with high resolution.
And you mentioned glycomap earlier, applying this to cancer cells.
Yes, that's a fantastic application. GlycoMap uses metabolically incorporated acidocyalic acids, basically, tagged sugars that get put onto cell surface proteins combined with an iridium catalyst. This allows researchers to precisely control the labeling of the sialylated proteome, which is often altered in cancer, right on the surface of living cancer cells.
Super targeted.
Very. Now, a general challenge for metal photocatalysts has been getting them to penetrate the cell membrane effectively. But cell-permeable options are being developed. For instance, using catalysts like acrylflavine, functionalized with a halo tag ligand, allows targeting to specific proteins inside living cells. These methods can profile PPIs within just a few nanometer radius, for example, looking at proteins interacting with histone H2B or specific RNA binding proteins. Overall, these photocatalytic methods offer potentially higher spatiotemporal resolution and more focused PPI profiling compared to many of the enzyme-catalyzed techniques.
Okay, that's fascinating. Finally, let's explore how unnatural amino acids, UAAs, are used for labeling. This sounds like the ultimate in precision.
It's certainly aiming for that. The principle here is really clever. You use genetic code expansion technology, basically, tricking the cell's machinery to incorporate UAAs that contain photocross linkers precisely at specific sites within your protein of interest.
So you're not just attaching something to the protein, you're building it into the protein sequence at a defined spot.
Exactly. Then you activate these UAAs with light. They generate highly reactive species that react only with molecules within a very, very limited range, essentially, direct contact.
Why is this method considered particularly accurate compared to the others?
Well, it's potentially much more accurate than enzyme or photocatalyst-mediated approaches because you precisely control where the reactive group is located on your bait protein at the specific UAA site. And the reaction range is incredibly short. This significantly reduces false positives from non-specific background labeling. You're really capturing immediate neighbors.
What types of UAAs are used to achieve this photocross linking?
Several types. The main ones contain functional groups like aromatic azides, which generate reactive nitrines upon UV light exposure, or benzophenones like benzoyl alphenylalanine or PBPA. This forms a diradical that reacts with CH bonds. PBPA was used early on to capture the EGF receptor interacting with GRB2 inside cells. There are improved halogenated versions now, too, and diazarines. These are often preferred because they can be activated with less damaging long-wave UV light, are generally more reactive, and produce nitrogen gas as a byproduct, which is less likely to cause side reactions. DZPK is an example used with chaperone proteins.
Are there more advanced versions?
Oh, yes. There are bifunctional UAAs, like BP-kine, which have both the photocross linker and another chemical handle, like an alkyne. This allows for post-cross-linking functionalization, like adding a biotin tag for pull-down using click chemistry. Others incorporate selenium elements, making the cross-link cleavable, which can help in analysis. Or combine features like diazarines, selenium, and a click handle to really minimize background.
This raises an important question. Just how specific can these methods get in terms of identifying the function of interactions?
They can get remarkably specific. A really powerful strategy called cytomics uses bifunctional UAAs containing both a group that mimics a post-translational modification, like lysine acylation, and a diazarine photocross linker. These were incorporated into histone proteins right on the chromatin.
Wow.
Yeah. So they could specifically capture interacting proteins that recognize those particular acylated lysines on the histone tail. This led to identifying a protein called GLYR1 and understanding its role in modulating where a specific histone mark, H3K56CR, is located along genes.
That's incredible detail. What are the limitations here? Is anything still non-specific?
Well, even with diazarines or benzophenones, the activated intermediates themselves are not usually amino acid specific in terms of what they react with on the prey protein. They'll grab onto nearby CH bonds mostly. This can still lead to some background if there are lots of proteins nearby. People are exploring residue-selective photocross linkers, like ONBAK derived from lysine, to try and address this, making the reaction target specific amino acids on the partner protein. And typically, these UAA methods are focused on the interactome of a single protein where you incorporate the UAA.
Right, one protein at a time.
But advancements are allowing for more global approaches. For example, using engineered tRNA synthetases to incorporate bifunctional UAAs with diazarine and alkyne globally in place of a natural amino acid like methionine. This allows profiling host protein interactomes during, say, a viral infection on a much greater proteome scale. Still challenging, but moving in that direction.
Okay, let's try to unpack this. We've covered an incredible amount of progress, truly amazing tools. But despite all these advances, what are the remaining hurdles? What are the big challenges still facing scientists trying to get a complete picture of PPIs in living cells?
That's a really important question to ask because, yes, the progress is stunning, but several significant drawbacks still need addressing. First, most of these sophisticated strategies still require genetic manipulation. You need to engineer the cells to express the exogenous enzymes or incorporate the UAAs.
Which limits their use.
Exactly. It limits their application in primary cells or patient-derived pathological samples, things that are often very difficult or impossible to engineer genetically. Second, there's the issue of enzyme size and steric hindrance we touched on. Many of the enzymes used, like BioID or even APEX2, are quite large. Fusing them to your protein of interest can physically get in the way. It might affect the protein's normal function, like its trafficking within the cell or its localization.
Even the split strategies.
Well, split strategies help mitigate this by using smaller tags initially, but they often require adding external small compounds for control, which adds another layer of complexity and potential perturbation. Third, and this is a big one for proximity labeling, distinguishing true direct interactors from the larger pool of merely proximal proteins.
Separating the actual conversation partners from the bystanders in the crowded room.
Precisely. Proximity labeling is robust at profiling the general neighborhood, but pinpointing the direct functional binding partners remains challenging. More effort is needed to effectively reduce that labeling radius even further, perhaps down to angstroms. And finally, complexity. While we can now characterize potential partners for one protein much better, analyzing multiple interactions within complex protein networks simultaneously under the same conditions in living cells, that remains a truly daunting task. It's a whole different scale of challenge.
So putting it all together, what does this all mean for the future of this field? Where are we heading?
Well, if we connect this to the bigger picture, I'd say the field, despite its rapid progress, is still really in its infancy in many ways. Given the absolutely central role of PPIs in nearly all aspects of cellular activity and disease, the ability to truly elucidate their complicated functions in their native contexts, that's a holy grail being intensively pursued worldwide. We can expect continuous interdisciplinary efforts, bringing together biologists, chemists, physicists, engineers, developing new, evolving technologies. The push will be for even greater spatial and temporal control, more sensitivity, less perturbation, enabling non-invasive exploration of PPIs not just in cells, but hopefully routinely in whole organisms, in animals.
The potential seems enormous.
It is, leading to fundamentally new discoveries about how life works and hopefully entirely new therapeutic strategies and applications.
So as you reflect on this incredibly intricate dance of proteins happening inside your cells right now, just consider this. Every breath you take, every single thought you form, every defense your body mounts against disease, it's all a symphony of countless coordinated protein interactions.
And what might be possible when we gain perfect, real-time clarity into this molecular choreography, not just understanding it passively, but maybe even subtly influencing it, guiding it with unprecedented precision for health.
We've given you a deep dive into the truly fascinating world of protein interactions and how we're learning to watch them. Keep that curiosity sparked. Because the future of understanding life, and perhaps even redesigning it for health, is literally unfolding right before us, molecule by molecule.